Okay, all right, thanks. Thanks for joining me here in the EP Blue studio. This is our remote demonstration system, and I would like to, since I'm not able to be there in person, to show you our system. Uh, Still working out. This is my first time here in this studio, so bear with me if there's a few technical bugs. We'll figure it out. Okay.
Okay.
Um, the uh, I should introduce myself. I really hadn't had much chance to talk with you previously. My name is Phil Hebert. I'm the instrument sales specialist for covering your area. I've been with Eppendorf for a long time, for about 20 years. In the last 11 years, I've primarily focusing just on sales of the automation system. So I'm pretty sure I can answer any question that you might possibly have related to our things. Let me ask you first, before I get started, what is the what brought you to us? Why did you think of Eppendorf when you were looking for an automated liquid handler?
So we have to do a lot of library prep. So that's why we are thinking about using a robotic system to run it. We have a couple of mistake when we training our new members to do the library prep. So we hope we can have a very stable performance on the library preparation.
Okay. Do you have a particular kit in mind? Is there some particular kit that you're using?
We now more focus on the Illumina DNA prep kit.
Illumina DNA preps. Okay, pretty
Yes.
much. That's good news. Eppendorf, we have optimized pretty much all of the Illumina kits. I think you'll see that they do recommend us for a great many of their kits, and the 5075T system, the one which is right here behind me, is the usual choice for that product, for that kit. Actually, from virtually all of the NGS library kits. Do you have any experience with Eppendorf?
Yeah, we have a couple machines from Eppendorf, like a tabletop centrifuge, and also we have incubators, Eppendorf incubators. So we pretty like the incubators because they have, automatic autoclave system, so sterilized system, so it's very easy to handle.
Have you ever used our pipettes?
Yeah, we have like a reference 2 in our lab, like 3 sets.
Okay.
I like it.
Okay.
Yep.
Okay. So your experience with those, how's your experience with Eppendorf been?
Pretty good.
Pretty good?
The product's
Okay.
great. It's great. Yeah.
I feel that's I feel safe asking that question. I've been asking that question to every customer I've ever demonstrated for years and years, and very rarely get any kind of negative response. I mean, granted, our products are not perfect. I mean, sometimes things need service, and occasionally things break even. But the reason why I feel comfortable is that the vast majority of customers, really have good experience with our products. They feel like they perform well, they hold up well over time, and that's a great reason. Thank you for considering Eppendorf for this acquisition. Do you have any experience with any other liquid handlers? Have you ever used any other types of liquid handlers, and what has that been like?
I haven't used it personally, but I saw people doing that on the Hamilton system.
Hamilton, okay.
Yep.
Did you get any feedback on that? Do you know any on what that was like to use?
A little bit complain that it's very hard to write a program, and also it's very expensive machine.
Okay.
So that's why
Good
we're
point.
thinking about Eppendorf.
Okay. Any other experience with automated liquid handling?
Not really.
Okay. Well, first let me tell you how I'm going to do this demonstration. A normal demonstration typically lasts about roughly an hour and a half, give or take, depending on how many questions that you have. Sometimes it runs a little bit longer. I normally do is I like to do an overview presentation where I will show you, there's an Excel I have a PowerPoint which has basically some key bullet points. You don't really have to pay too much attention to that. It's just to keep me make sure that I don't forget to cover every topic of interest to you. And it's going to cover a little bit of everything, all the features and benefits of the workstation, why this is the best choice for your application, care and maintenance. Then normally after that, I'll do a little bit of a show and tell. In the process of doing that, I'll do a show and tell where I'll actually take out physical items and show you how they work. Then I will open up the software, write a couple programs for you. The first one will be extremely simple and just take a few minutes to write, but I will explain to you why I'm choosing the settings that I'm choosing when I write that program. And then we will simulate it, run it on the machine. All of that should only take maybe 10 minutes or so. Once I'm done with that first one, I will write a somewhat more complex application that will use more features. So you'll be able to see the machine move things around, heat, cool things, shake things like that. You'll have a chance to see more, get a broader view of how the machine works and actually see the speed and that type of thing. After that, I will be happy to answer any questions that you have. But by all means, speak up instantly as soon as you have a question. At any point in time, I say something that doesn't make sense to you, let me know and I'll clarify it for you, and we'll be back on track. Okay.
Okay, sounds great.
I'm going to go ahead and start over here on the monitor. So if you could cut to the monitor, and I will. I'm here really to answer that question right there. Why should you buy the epMotion system from Eppendorf? These are, Eppendorf has been a world leader in laboratory technology for generations. Everybody that goes to buy a workstation wants to know they're buying from a reputable company that's going to be around for a while. Well, Eppendorf has been in business as a privately held company for over 75 years. All of our products are engineered and manufactured in Germany, a few items here in the United States, to meet the most demanding specifications. You may not be aware, but Eppendorf, has innovated many of the things you use in the lab every day. I don't even know if you may not even realize it, but we have the patent on the very first piston stroke pipette that has ever been that was ever put on the market. The microliter system, the Eppendorf tube, an Eppendorf innovation, the first DNA grade microcentrifuge, and many people will tell us those things last for decades. I personally have a customer at UNC that had one that was that very first model that was produced. It was 42 years old and still chugging along. Eppendorf, the flame photometer, predecessor to the modern spectrophotometer, was an Eppendorf patent. We also have the first femtoliter cell micro-injection system, and we patented the gradient PCR machine. So we're not new to this business. I think we make a lot of products that are very, very highly regarded in the industry and have been doing it for a very, very long time. We're consistently ranked #1 internationally in product satisfaction and customer loyalty among all product suppliers to the life science community. So we have been doing this. I think you should feel comfortable dealing with a company like Eppendorf. Anytime people buy a large workstation, they also want to know, what is your service organization like? How are you able to help us? Well, we are I feel that Eppendorf takes better care of customers primarily because we sell so many products. We have a catalog, a couple inch thick. Although granted, it hasn't been put into print this year for the first time. It is fully online. A lot of our products are sold through distribution channels such as Fisher Scientific and VWR and things like that. And we were actually ranked 10 consecutive years in a row as the #1 field support and service for all the companies that they represent. We had an internal survey back in 2020 where customers were asked about their satisfaction with our service, and 98% rated us as satisfied or very satisfied. So we can take care of you. Now, our workstations are serviced for the most part, entirely on site. We are not one of those kind of companies that's going to ask you to to ship your workstation back to a foreign country. We're not going to tell you to throw it away. This may shock you, but there's actually a company out there who doesn't service their instruments at all. And once it breaks, it's outside of warranty. If it breaks inside of warranty, they'll give you a new one in that first year. But after that, if it breaks, it's like throw it away and buy another one. I'm not kidding you. We don't do it. We're not that kind of company, okay? We have a team of dedicated professionals to help you. You have a local Eppendorf rep that is based locally to you. I'm an automation specialist, so I focus primarily just on automation. I have a much larger territory, but I can get to anywhere in my territory, usually the same week or the following week. So if you ever require support or assistance and you need me to be on site, just let me know, and I'll try to work you into my next visit. Usually, customers don't have to wait more than a few days or so. We also have an application support team, an entire group of people called our BioApps team, who is there all day to help you with any kind of problems that you might have. You encounter something you don't know how to do, you can reach out to BioApps at eppendorf.com. They will respond before the end of the business day with answers and try to help you through whatever problems you might be having, whether it's a problem with our instrument or not. Let's get to what makes the epMotion system unique. I'm going to be talking about some of these things here in this on this page. The flexibility of our workstation, the accuracy of our workstation, the programming ease of use, our patented optical sensor, the ability to do free-jet dispensing, and the unique reliability of our workstation is in a class by itself. And so I want to share some of these. I do have a slide for each one of these things, so I will not expound on it right here and now. Now, the first question that any customer wants to know is can you handle the application that I have in mind for our workstation? In order to answer that question back, I only have to ask a few questions back to a customer back. Our instrument has a pipetting range of 200 nanoliters to 1,000 microliters in 4 pipetting ranges with a lot of overlap there. We have 1 single and 8 channel tools. There are 0.2 to 10, 1 to 50, 20 to 300, and 40 to 1,000 microliter tools. If you could imagine that you have a set of single and 8 channel pipettes in those ranges. If you can perform your application by hand with a set of single and 8 channel pipette in the thing, then we can do it as well, because our machine exactly replicates the way you would do it by hand on the bench. There are only a couple of caveats there. Our machine can pipette into or out of any brand of style tube or plate. It doesn't matter. We don't require you to use our plates. We don't require you to use our tubes. We don't require you to use anything specific except for one thing, reservoirs. No one else makes a reservoir quite like ours, but there are other options besides our reservoirs. And also, matching tips, the proper tip size. You do have to have the proper tip size. Our workstations can incorporate a wide variety of different things: 2-dimensional shaking, vacuum extraction, magnetic bead extraction. All of our systems, except for the 5070, have the option for a UV HEPA clean cap, so you can keep the interior of the machine clean and sterile and nuclease free. Our machines also allow for the reuse of tips for people who do not have sensitive applications and can reuse tips over and again. That's something that can easily be programmed into a program. So let me show you let me show you. I'm going to go try to get this into the view here. This here is I'm not really that used to this video on this angle. So Oh yeah, thanks for zooming out there. Appreciate that. This here is a reservoir rack that I mentioned. It includes a variety of different types of adapters. We make adapters that fit into the reservoir rack that can hold virtually all common tube types, 0.2, 0.5, 1.5, 2 ml tubes, 15 mil conicals, all the way up to 50 mil conicals on the high end can fit inside adapters that fit in here. And we even have modules that can hold tips and other types of things. So you can mix and match whatever it is that you need for your application simply by putting them into the proper places and into places in here. And then you'll tell the machine how you've set up this rack and then use it as you want. Does that make sense? Oh, I'm sorry. You may have muted it from the background noise. That's okay. I see you nodding. This is a very flexible system. It's really easy to use. You just like I said, mix and match, and like I said, we have something for everything. We also can use reservoirs. We also have reservoirs, high volume reservoirs like this 100 ml reservoir, mid volumes like the 30 ml reservoir, and our smallest ones, the 10 ml reservoir. The only rule here, of course, is to use you can only use a reservoir that the tips can actually reach the bottom. So you wouldn't be able to use this, for example, with anything other than the 1000 microliter tool, because the only tips will go to the bottom there. So other than that, now our machine can pipette into or out of anything you can imagine that's shorter than a 50 mil conical tube sitting inside of this rack. Even if we don't have something custom made for it, even if we have to make it custom, we can do that. Okay, we can pipe it into it. Now, the machine does have to be able to access it directly from above or at the 90-degree angle to go directly into the to go into the thing. And the other limitation of our machine is that the machine cannot automate something that requires manipulation of the item in the course of the run. For example, our machine cannot remove a cap in the mid run or anything like that. It can't do that kind of thing. You would remove them at the beginning of the run. So just to recap, anything that you want to go into or out of, heat or chill, shake, that is the appropriate weight for shaking. Anything shorter than this, accessible from a top, tips can fit into it, not too tall, we can work with you. I don't.
Recently, we have colleagues that are trying to run the ddPCR plate, but the ddPCR plates are very unique. They are not like the 96 well plate. Are you able to design
yes,
to run
yeah. Yes, actually, we do have we've had customers request that before, and so we do have an adapter for ddPCR plates. We can make, even if we don't have it listed in our catalog, we can make it for you. I have seen some incredibly complex adapters made. One example, I had a customer who wanted to pipette into slides. We laid the slides. It turns out we could lay out the slides, but it would take 6 deck positions. So we had to create a custom rack that would slide, where you could slide in the entire thing. The whole thing slid into the workstation, covering 6 deck positions, and then we picked in the individual spots in the slides. We can even do that. Okay. I had another customer who had a really, I thought it was sort of insane request. They received tubes from all these different people, it was a simple application. They just had to take 50 microliters out of each one of those blood tubes and put them into a thing. That sounded easy enough, but the problem is that they didn't want to mix and match anything. They just wanted to be able to put them all in there any old way. So we created a custom nested thing where it had holders that could. It had springs on the side that pushed each individual tube, centered the tube, so no matter whether it was small or large, it would always stay in the center, and it would also fall to the appropriate depth so that we could pipette into it. It was pretty cool. I was like, when I got it, I was like, oh, I can't believe we can do this. This is like blew my mind. It's fun to do these custom things. So if you can dream up something, we can find a way to do it. We will find a way to do it. Okay, you won't need to worry about that for NGS, but you may think about other applications here in as we go along that you would also like to automate. So I'll talk to you a little bit about that now. Okay. I'm going to cut. If you could, we could cut back here to the screen. Okay, this is a fairly exhaustive list of of applications. Let me see if I can get this a little bit more centered. Fairly exhaustive list of applications. Our machine can do basic liquid handling, tubes to plates, reformatting, 96 to 384 wells. If you haven't done 384 well qPCR, sometimes people consider that to be a difficult thing to applicate, something we do very, very well. Our machine is very well suited for doing it, going into 384 well plates. I've even done 1536 myself, although our applications lab didn't like me very much because technically that's outside of our specifications. We can do normalizing. Do you have to normalize your samples? Have you ever tried doing that?
Yep.
Yeah.
Yep.
Well, I mean, I think that falls in the category of something that is so complicated that nobody in their right mind would want to do that by hand, right? You have to pick a different volume from a different location and go into a different well each time. You got to use a different pipette sometimes. You got to reset your pipette every single time. It's mind-numbing. I had people tell me that just doing one 384 well plate would wipe them out for the entire day. Well, I'm here to tell you that on our machine, once you write a little template, you can use an Excel spreadsheet, and the machine can take the data and directly produce the entire protocol for you. I've seen it. I've plugged in a spreadsheet that had 384 individual picks from all the wells of a 384 well plate with different volumes and all, and it can write that. It will write that program literally in about a couple clicks. You click on import from CSV. You click on the command that's going to be your template, and then you click on import from CSV, and boom, you've got 384 individual picks all programmed. Different volumes, different locations, different sources, different destinations. It doesn't matter.
Wow,
you can
even
do
a lot of time.
Yeah, you can even do multiple plates. You can pick from multiple plates, tubes, whatever you want into whatever you want. So it's that easy. So I'm happy to teach you how to do that if you decide to get one of our workstations. A very similar application, hit picking and pooling, where you have to pick individual ones, same kind of thing. Sterile cell assays. We've had people that do cytotoxicity assays, viability and apoptosis assays, media exchange, things like that. The UV HEPA clean cap helps with that. The UV can irradiate and sterilize the deck. You can chemically sterilize the deck if you wish to. We do recommend 70% ethanol. If you use bleach, we recommend that you follow it with ethanol in order to wipe away any residue. Our PCR setup was one of the original reasons why we introduced the epMotion. Because no company had really dealt with precision and accuracy at really low volumes before our instrument was introduced. So it was a uniquely good thing. You're going to be. Do you do any quantitative PCR in your lab?
Yep. Do a lot of qPCR.
The good news there, one good news there is that many times people can even pay for the entire workstation just with the savings that they experience from reagents alone. To give you an example, like setting up a 384 well plate, doing it by hand, most people would do that application with substantially less than, they usually want to do 10, 20 microliter reaction volumes, whereas this machine can pipette so much more precisely that we can get down to a much smaller size. I have customers that do 4 microliter total reaction volumes and get a great result, better than they could do by hand with 3 times that volume.
They save a lot of money for that.
It can over time. I mean, that can save right there hundreds of dollars a plate, and if you're doing it a lot, then you can pay for the machine in a short time.
Okay.
Nucleic acid purification. We have optimized a huge range of different types of kits on our machine. Mostly are mag beads, but some of them are also filter plate extractions, plasmid preps, and so on, as well as protein work, ELISA setups, and stuff like that. So it's a long list of applications. It's not all inclusive, but it does thing. Do you have any? You see anything on that list? I guess you see a few things on that list you might be able to incorporate.
I think that's we basically do QRT PCR and kind of cell culturing. I think you can do it like cell seeding, those kind of things.
Yeah.
We also try to do transformation. Is that possible?
The liquid handling portion of it, obviously, there's no. I mean, if you're talking about electroporation or anything like that, we don't have any kind of electroporation.
No,
I'm not sure
just
specifically what that.
chemical transformation,
Okay.
like lipoprotein transformation.
I'm a little hazy on that. Not something that I've been asked about really, but I could get back to you about that. I could get it back to you about that because I'm not really that familiar with that particular application.
No problem.